So hello, and thank you for the presentation. I'm Henri Barry, a third-year PhD student in the physics lab, and I will talk to you about the impact of the chemical modification of antisense oligonucleotides on their interaction with the target. In France, in the year 2000, we had a national health campaign that was antibiotics are not automatic. Why? Well, because of antibioresistance. Antibioresistance is a really important subject. It is estimated that by 2050, it could be 39 million deaths. So it's a really important subject, and we are running out of new classes of antibiotics. So our strategy is to use the antisense strategy on resistance gene of bacteria. So most of you know already the antisense strategy. One point with the antisense strategy, we use short oligonucleotides. So we can modify them quite easily and bring a lot of chemistry to it. So the antisense oligonucleotide binds to the messenger RNA, and then the ribosome is not able to translate the resistance protein in our case. The oligonucleotides that we use in our lab are modified usually with this lipid conjugate. So we see that it's actually a nucleolipid where you have the nucleotide core with two lipid tails. This gives the oligonucleotide the ability to self-assemble into micelles and also the ability to concentrate in cells. Here you have two oligonucleotides, one without the lipid and one with the lipid, and you can see that in bacteria, it actually concentrates on the bacteria. Here you can see the micelles observed in a 10 microscope. So one of the main challenges with working with oligonucleotides and bacteria is that you have to enter the bacteria. And you don't have endocytosis in bacteria, and you have two membranes in gram-negative bacteria. So it's a really big challenge, and it's a real pull that we have this nucleolipid to enter bacteria. So our target is the messenger RNA, and this messenger RNA is usually translated into a resistance protein. In our case, it's a protein that degrades beta-lactam antibiotic. I talked about the chemical modification. You have seen the first chemical modification, which is the nucleolipid, but we can also make other chemical modification to increase the ability of the oligo to be used as a drug. So those kinds of modification, usually you know the PTO modification and also the ribose modification, which all improve the resistance against nucleases and increase the lipophilicity, which is important if you want to enter cells. So what are the questions that I'm asking on this project? So how does the lipid oligonucleotide interact with its target messenger RNA? What is the KD of the interaction? What is the melting temperature of the duplex? And how impactful are the chemical modification on this KD and melting temperature? And also, what does the conformation does the duplex stage? To do this, I'm going to use isothermal digestion calorimetry. Now everyone is familiar with it with the last talk. So isothermal digestion calorimetry is great because you don't need to use a probe, and you can do it in solution in native condition. You can do it in any buffer you want, as long as the buffer match with the sample that you use. But the downside is that the acquisition can be quite long, depending on your condition. But you get a lot of parameters from isothermal digestion calorimetry. So these are the results that we have with our oligos. So the oligonucleotide is just the one that we use, and then I have synthesized the messenger RNA matching this oligo, just the short pair, the 28 base that we use. And you can see that when we have the lipid, the interaction is less strong. It's expected because it's a big molecule that you add at the end of the oligo, and it's actually not preferential. But you still have KD that are in the nanomolar range, so it's pretty good for our application. And if we do this for every modification, so the first one is the oligonucleotide of DNA, so not modified, and the one that you saw right before, then we do two or methyl modification. The KD are about the same. What's pretty strange is that with LNAs, we see that the KDs are much higher. So I don't really know why we see this, especially that with the melting temperature, it's not the case. So this one I'm not really sure. Maybe the data analysis of the ITC is not exactly the same. Some parameters need to be tweaked. But with the MOE modification, it's where we have the best results. So even with the nucleolipid in the orange bars, you can see that it's pretty low. So even with the nucleolipid, which is not favorable for the interaction, we still have a pretty strong interaction. We also have the same thing, but without the modification. And those are the binding profiles. So the binding profiles are the data you get from the ITC. You have the delta G, which is representative of the KD, and also the contribution of the enthalpy and entropy. This interaction is obviously enthalpy driven and not entropy driven. The binding profile that we observe are the same for oligos. I had a hope that we could see some differences depending on the modification, but that's not the case. All of the binding profiles are the same. We also did circular decreasing to learn more about the structure of the duplex that we form. The structure of the duplex of oligos can be really important, especially if you want enzymes to recognize the oligos. For example, RNase H does not recognize A form or B form duplex the same. So we did circular decreasing to better understand the structure, and you can also get melting curves from circular decreasing. So the first thing we did is the melting curve, and you can see that with this 25 base pair oligo, all of the melting temperature are above 50 degrees, so it's a really strong interaction. You can also see that depending on modification, you have to increase a lot the melting temperature. And the PTO modification is not favorable for the interaction. So it usually lowers the melting temperature by about 4 degrees. Now, if we look at the structure of single-stranded oligos in solution, what you see here is that single-stranded oligonucleotides without the lipid are present in two forms. You have the RNA that is in one form and the oligonucleotides that are in one form. With the lipid oligonucleotides, they are all in the same form, this the form of the RNA. When you add RNA in the solution to form the duplex, everything shifts to the RNA into the B form. That's because RNA is more rigid, so it forces the conformation of the duplex into one form. But with the lipid, it's already in this conformation. So that's an interesting result. We did this with all of the modification, and what we see is that the more modified the oligo is, the closer to the RNA it is, the closer to the structure of the RNA it is. When you add the RNA in the solution, everything shifts to the RNA and everything is in the same form of the duplex. So the conclusion are that the nuclear lipid increases the KD, so it's not good for the interaction, but it's necessary if we want to see an effect in bacteria because they don't penetrate the cells. The PTO increases the KD also, but it's also necessary because it confers resistance against nucleosides. But with 2 MOE modification, you lower the KD and you basically recuperate all of the interaction. So modification can be really important if you want to work on bacteria and if you don't want to use the PMAs. With the structure insight, we see that we have 2 different families with lambda-max presented right before also on the cycle of the crescent, with 263 nanometers lambda-max or 280 nanometer lambda-max, more closely related to DNA. And you see that the RNA binding forces the conformation at 263 nanometers. So I'd like to thank the ARNA for the funding of this project, Dr. Cameron Maturans for the training and the ITC, my institute students for the DLS measurements, and the opto-oligosynthesis platform with Brunelli and Patricia Gorbach for the synthesis of oligonucleotides, and the Campbell Foundation, and you for your attention.